In this lecture, I'll be going through Introduction to Genetics. So first I'm going to explain the different terms that we're actually going to use in this subject. Yes. So first thing we're going to talk about is character. So character, okay, actually I have to talk about character and trait at the same time. So character is, you can think of character as a bigger category. For example, the category is eye color. Okay, and trait will be blue eye, brown eye, or I have no idea what other eyes are there. Gray eye, for example. Yes. So for example, character is like what kind of like features you're going to show, like that is going to be present in everyone. So and trait is going to be the version that you're going to have about it. Okay. All right. So next, we're going to talk about gene. So, okay, so gene is no, it's not from X-Men. Gene is about is a fragment of, you can think of, okay, not fragment of DNA. It's like a portion of DNA that codes for one trait. Okay. So think, okay, for example, if we have a DNA strand that is as this long, then for example this part is the gene that codes for the eye color. So it codes for the enzyme. So when this is expressed, it codes for the enzyme that catalyzes the reaction to form the color pigments in your eyes, for example. So when we talk about gene, it's literally DNA that codes for a single protein, which is a single trait. Okay, next is we're going to talk about homozygous and heterozygous. Okay, homozygous means that the two alleles we're going to have. Okay, actually I have to go for allele first. Yes, allele. Okay, for allele, right? We can think of it as a version of gene. So, for example, like actually I said it in the previous video. For example, if the gene is coding for eye color, so one allele we can have is AAATCCG, and another type of allele we can have is AAAGCCCG. Okay. So in other words, there was a single. So here that okay. So here are the three amino acids that it's going to code for. So in other words, the amino acid that's been coded from this code on this from this coder is going to be different from this one. In other words, for example, this one is going to I'm not saying that it is right, I'm just giving an example. This one is for valine, and for this one is for glutamic acid, for example. I'm not saying that it will, it's like, I'm just giving an example. We call it example. Yes, where was I? Yes. Yes, then the final structure of the protein will be different because these amino acids will be having different type of interactions with other amino acids within the protein. So it's going to catalyze different kinds of reactions. So in other words, when we talk about allele, right? So it's going to they're going to code for the same enzyme, but serves a slightly different purpose. For example, for this guy, well, let's say this guy is brown. Okay, then this guy is going to have an enzyme that is going to that catalyzes a reaction that forms the brown pigments. But for this guy, he's going to the enzyme is going to catalyze the reaction that forms the blue pigments. Okay, so it's literally allele is like a different type of gene that we can have. And when we talk, okay, so and usually for this one we we annotate them as with alphabets like and A and B, like A, big A, big A's, like big A, small A, small a, or it can be just big Y, small y, and so on and so forth. It can be literally it can be any alphabet, but my suggestion is don't use vague ones like C and C because it's pretty easy to see here, but when you actually write it on paper, people might be really confused, like really confused. Okay, so when we talk about homozygous means that the two alleles we're going to have is going to be the same. So for example, in this case, the big A and small a is going to be homozygous. And in the case of heterozygous, it means that the two alleles that this person has is going to be different. Okay, the reason why we have heterozygous and those kind of things is, right, if you recall, we have two pairs of chromosomes for each chromosome number. For example, for chromosome number 1, we're going to have one from your father, one from your mother. Okay, although this looks like chromosome, but in reality, recall that it doesn't say in chromosome, it stays in chromatin, but for the ease of showing, I'll show in chromosome, okay? Remember, it's during interphase, it's always chromatin. Yes, chromatin. Yes, what was that? I was carried away. Yes, so heterozygous means that the two alleles are going. Okay, it's homozygous. For example, this allele is going to, for example, is big A, and this guy is also big A. Then this guy is homozygous. But if this person is going to have big A and small a, means that this person is heterozygous. Okay, and then we can actually bring the dominant allele and recessive allele in here. Okay, usually for dominant allele, we show it with capital letter. Means that if we have, okay, we're going to talk about heterozygous. In the case of heterozygous, the effect of A is going to mask the effect of the small a. In other words, when the big A is present and the small a will not be will not be expressed. Okay, so for example, let's say the allele big A codes for brown eye, and the small a codes for blue eye. So if this person has the allele big A and small a, the small a will not be expressed, and only the big one will be expressed. So if the person is heterozygous in the eye color, this person is going to have brown eyes. And by the way, just for a tip, brown eye is the most dominant eye color, followed by blue, gray, green, and purple. Yes, they are actually purple, which is really rare. Yes, I'll carry on to the next one. And genotype, yes. So actually, I also have to talk about phenotype as well. Oops. Okay, phenotype. So phenotype is what you see. So it's the result of genotype. So it's something that you see with your naked eye. So it's a trait that is being expressed outside. So you can think of phenotype as something like what you see as the character and trait. Okay. So for example, the eye color is the phenotype. Actually, I should say the eye color being blue is the phenotype. And the genotype for this is going to be homozygous recessive of small a and small a. So this is a genotype, which means it shows the genetic makeup of this person, and the phenotype shows the physical trait that has been expressed from this genotype. Recall that this is also DNA sequence, and when this is expressed, it becomes we will get a protein product, which is going to be which is literally what our body is made of, like the colors or whichever feature that we have. Okay. Okay, so sex linkage means that the allele that we're concerned with is going to be in the sex chromosomes. I remember that there are there's major of two types of chromosomes we have. One is the sex chromosome, and one is autosomal chromosome. So autosomal means that it's from for human body is from chromosome 1 all to chromosome 22, and sex chromosome is going to be your chromosome X and Y. Well, for girls you only have this. For guys, yes, we have this Y, which is yes. So in other words, if the allele is on either chromosome X or chromosome Y, yes, it is sex-linked. Sometimes we usually call it X-linked because most of the alleles we're concerned of is found on X chromosome and not on Y chromosome. And usually it is X-linked recessive because X-linked dominant means that literally everyone will have it. And the reason why it's not found on Y is because if the length of X chromosome is this long, Y chromosome is really like that long, which is really short. Like, so now we're going to talk about non-recombinant gamete, which is also parental gamete, and recombinant gamete. So in this case, we have to talk about linkage first. So in other words, remember, sex linkage means that the allele is on the X chromosome. For just the normal linkage that we're going to talk about is it can happen in any chromosome, and in this case, it means that the two alleles. For example, let's say we have allele A and allele B, and they're going to be on the same chromosome. Okay, so the white and the blue one is going to be A, and the red one is going to be B. So, and since this person has two chromosomes, remember, all the individuals will have two pairs of chromosomes, one pair of chromosomes. So small a and small b. So this is what this person is going to have, for example. Okay, so this person is currently heterozygous. So if I write this down, this person's genotype, yes, it's going to be, okay, so what this means is that this big A and big B means that it's going to be on one chromosome, and for a small a and small b, it's going to be on the same chromosome number but another chromosome, and like this. So let's take a look at what happens to this meiosis first. Okay, remember that first the homologous chromosomes separate. So we're going to have big A, big B. To have small a, small b, and when this guy split again, we're going to have two of big A, big A, big B, and two of small a and small b. Okay, so these are the things that we're going to get. Okay, but for example, in this new scenario, we're going to have crossover and recombination. If you can't recall what crossover, over, and recombination is, please go through the previous notes and lectures. Yes, I truly encourage you to do that. Okay, so we're going to close up. Oops. Okay. So we're going to close up at this area. So, okay. So this person, this one will be, okay. Oh, just for information, just in case you don't know, recall that this one has been duplicated. So in other words, this one will also be small a. This one is also going to be small b. Okay. In other words, okay. So if we zoom this in. The one on the right with the small a, and the one on the big A at the right with the big A, and will actually have, okay, and if there's a crossing over here, forming chiasmata here, and if it breaks, this will lead to having small a at the left and big A at the right. So the final product of this recombination is, oops, okay, we're going to have is going to be like this: big A, small a, big B, big B, big A, small b, small b, small a. So in other words, if we separate this again, the homologous chromosomes, then into the sister chromatins later on, we're going to have 4 different kinds of gametes and these are going to be big A, big B, small a, big B, big A, small b, small a, and small b. Now, so this is the result of the linked genes, and the thing to know is that the crossing over actually does not happen all the time. So in other words, remember the independent assortment? Yes, it can happen both at this similar chances. So in other words, that if they're on different chromosomes, the chances of having the big A, big B, big A, small a, and big A. I'll just write that down. The chances of us having big A, big B, big A, small b, small a, big B, small a, small b, having these four will have the same chances if they're on different chromosomes. But in the case of linked genes, the fact is that the crossing over is something that may happen or may not happen, and usually the case you can assume that it doesn't happen. In the syllabus, as what I said, we assume that per meiosis there's only one crossover that occurs. In other words, this recombination would is actually unlikely to happen. Therefore these two are actually the rarer kind of gametes that we're actually going to have. Okay, so if I link this back to the terms, non-recombinant gamete, or which is also called as parent gamete, is going to be this one, the one I'm squaring with blue, because it is the same as what the parent originally had. But in the case of recombinant gamete, is the one that I have square and rectangled it with green, because it is the one that is different, the one that has been, it's like a new, how should you say, new type of genotype, yeah, that arose from the recombination of these the crossing over and recombination. Okay, so I'll just add on to the recombination effect of recombination. Okay, the reason why we have recombination frequency is to check that how like among the sample size, how many of recombinant gametes or offspring there are. So in other words, what are the chances of having recombination in this case? So this so the calculation for this is number of recombinants plus minus root of number of recombinants divided by total sample size. So for example, if there's 1,000 of them and we have 100, let's go with the 100. Okay, so this is going to be our percentage, and this is going to give our error term. Our error term is there because there are possibility of errors within, like, because for example, the ratio are we getting for like A and B should be, for example, 1:1. So instead of getting 50%, I get something less in the case. For example, just by chance, it just happened to be that way. So it takes account into that. Oh, but of course, in this case, we won't get 50% because okay, I'll explain that later on. Okay, so recombination and frequency also tells us the distance between the genes. Okay, the reason being is that if we have a chromosome that is this long, and if we have the allele A here and allele B here, okay, I'll label opposite, but never mind. So this is scenario 1, and in scenario 2, we're going to have the same chromosome where allele A is located here and allele B is located here. Okay, so the distance between the alleles shows the chances of crossing over and recombination occurring. So the longer the distance is, the more DNA sequence, right? The more the DNA sequence, the more areas that crossing over and recombination can occur. So in other words, it's case 1 is going to have higher chances and more the more of recombinant gametes that it's going to have, because this one has much less area that can occur compared to case 1. So usually this recombinant frequency is also used to find the distance between the two genes. Okay, so for example, if the recombination frequency I'm getting is 31.8% plus 3.2%, for example. Okay, so this can be directly translated into 1.8 centimorgan plus 3.2 plus minus centimorgan. Okay, centimorgan is like an arbitrary unit. So so 1 centimorgan can actually mean like 100 base pairs or 10,000 base pairs, or whichever that fits the case. And oh yes, so about I'll get back to this restriction. Oh my goodness, recombination frequency percentage. Okay, so in the case of 50%, it is either, well, it's unlikely to happen, but it's if the chromosome like this, they'll located at the right opposite end. But this is one possibility. But when people usually say it is 50%, we're going to talk about these two will be unlinked, where if there's chromosome 1 and chromosome 2, which are different, allele A is going to be here, allele B is going to be here. So in other words, there's 50 chances of A and B happening. So it is good. So 50% means that it is unlinked. Okay. So in the case of 0%, 0% means that these two genes are like linked together, like really tightly. So in other words, A and B are going to be like next to each other, or it may even be like the same gene if it's like literally 0%, because there's no way that we can actually separate these two if they are the same gene itself. Yes. Okay. So now we're going to talk about the usual case we usually talk about when it's linked. It's the case whereby the percentage is between 0% to 50%. So this would actually mean this will show the distance between the two genes. And after we find the percentage, we can go to find the distance between the genes. Just one tips on the questions to find the distance between the genes. For example, A and B is 20, I don't know, and for example B to C is 15, and A to C is 5. Yeah. Okay, so in this kind of case where it's actually best to have the longest one first. So in the case, the one between A and B is the longest, so this 20. Then we do this accordingly. So B and C, from B to C is going to be 15. So it can be either here, or it can be like down the road. It can be somewhere down here. Then we talk about A and C is going to be 5. So it's going to be either here or here. And it seems that a C it matches the both places. So we put the C inside. So this is how we find the arrangement of genes within the DNA. So now we're going to talk about Testcross. Okay, Testcross is means to cross something with homozygous recessive. Okay, for example, if this person has allele big A, small a, big B, and let's say the smaller ones are the more recessive ones, we're going to cross it with small a, small b. Okay, so this is called a Testcross. So why we do Testcross is to it's actually to it's one way we use it is to check what genotype discipline is when this is actually unknown to us. It can also be used to check whether this is whether it's sex linkage or there's linkage in the first place, whether it is autosomal or sex linked, or whichever the case is. Yes. Mutation. Okay, mutation is change in base pairs. Okay, so actually there are different kinds of mutation. There are base pair deletion, there are base pair substitution, so on and so forth. But in this case, I'll just tell you about base pair substitution in the moment. Okay, so for example, if the person has DNA sequence that is like AATCGG, mutation can be something like the C is replaced by let's take a T. Okay, so in this case, there is a mutation that has occurred. Okay, so the original was CGG. Okay, so this can actually be, this may be lethal, this may not be lethal, and the thing is, we don't know what will happen. The actual fact about mutation is that it actually happens quite a number of times, but the thing is, we have a check off at the end of S phase of cell phase that screens something like screens the difference in nucleotides. So it's like there's an enzyme that checks whether the DNA has been properly duplicated, and it actually matches the original one. So if there's actually a change, it will try to remove the change and replace back to the what is correct. But as you know, we're not perfect, and so is our body. So sometimes they just miss this thing, and this one thing will carry on. This thing can actually lead to different proteins. For example, these will code for different amino acids, right? For example, if TGG codes for a totally different protein, we don't know what will happen. Well, actually, we can anticipate something like, but we won't exactly know just by looking at the nucleotide. But also, it can actually be the same as well. For example, if TGG codes for the I'm not saying the if it codes for the amino acid valine, and CGG also codes for the amino acid valine, then it becomes a silent mutation where there's technically it's a change that is good as none. And there's actually really interesting studies that actually makes use of this silent mutation to seek the evidence of evolution and so on and so forth. So okay, so these are the terms that is. Okay, so I'm going to teach you how to write the genotype and phenotype calculation in a way, and the Punnett square. Okay, so I'll use the example that is given by the school. Okay, so actually the first thing that you should do is actually state which allele that you're actually using. For example, in this case, I'm going to say the big capital R is for, in this case, I'm going to use it to represent dominant allele, and for which codes for round seeds. Okay, we're going to talk about PEA as well. Yeah. Okay. And for example, the small r, which represents recessive allele, which codes for wrinkled seeds. Okay. And for example, the capital Y for yellow color, which is dominant, and small y, which is going to represent, be recessive and code for green beans. Okay. Okay. So the next thing you have to state is that parental